And also one of my supervisors for my master degree is also Xi-Qiao's supervisor for master and the PhD. So Xi-Qiao is my younger academic brother. Okay.
No.
So let's start the interview. So Xi-Qiao, your name is Xi-Qiao means the West Bridge. Is there any reason for your parents to give you that name?
In fact, my name is given by my grandfather.
Okay.
In fact, I live in Hebei provinces. Before I came back to Beijing, I have never seen any bridges. In fact, because we live in the in the Hebei, we don't have river, we don't have lakes, and don't we have bridges. But during that time, that is the cultural revolution. My grandfather said that we should learn from Western countries. So all of my brothers use the second word West.
Okay.
My name is West Bridge, and my younger brother named West Xi-Qiao, 车轴的轴.
Okay.
And he's also a student in our department.
Okay.
Oh,
your
department,
brother. Engineer mechanics.
Your brother.
My younger brother. He studied in the group of Professor Feng Guo先生.
Okay.
The group.
Okay.
Okay.
And in fact, we have two other families also studied in the same department. And the wife of my brother is a student of Professor in fluid mechanics.
Okay.
And another one is also a student in the thermodynamics in our department. So we have four persons in my family graduate from this department.
So I'm very glad to hear that, because once upon a time I was the department chair. And so that raises my second question. So why you choose mechanics as the specialties of your university and later on the PhD subjects?
And first, I came from a very, very small village in Hebei provinces. In that village, we have only maybe 50 families, about 200 persons. And I studied in a very small school, very small high schools. In that schools, we have never any persons and people who can enter Beijing University, Tsinghua University, and such on. I am the only one in the past half decades. And so we know nothing. But I'm very good in mathematics. And when I was in the high school, I got an award of national competition of mathematics. So the school and many teachers told me that you must learn mathematics. But my father said mathematics is too difficult because I will know the 哥德巴赫猜想. It's too difficult. You should learn something related to engineering. So my father chose the department of engineering mechanics. That's why.
Oh, your father chose for you. Okay. And I saw the Sulin Zhang is now raising hand. Hello,
okay,
Sulin.
I'll promote. You guys continue.
Okay. So from your CV, we can see you went to Germany for a period of time with Professor Gross.
Yes.
And you also stayed in US for a certain period of time. So
With
if you
Yang Huang.
compare with the research style in continental Europe and the United States, what are the different approach they use?
Yes, I studied and worked in Germany, in the Netherlands, and in the US. And the Germany and the US are greatly different. My host professor in Germany, Professor Dietmar Gross, never told me that to do anything. You can do anything you like. And they can do a subject for maybe 20, 30, or even a whole life. For example, the micromechanics. Professor Gross is very good in that field. But when I went to UIUC with Yang Huang, he called us every day, even in the evening. Yeah, they push us very much.
So Yang Huang is here? Okay. So you are a very terrible host.
In the weekend, in the evening of the weekends, and Bin Liu Bin, Shao Xingqu, and many others, we play cards. We play cards in their home. And in a moment, Yang Huang called us. How is the equations are derived? Showing side, we are deriving. We'll give you tomorrow. Even in the midnight, Yang Huang is asking the equations. That's greatly different. And the US, we also always doing something in the frontiers of mechanics. But the European professors are very
Relaxed.
yeah.
Relaxed.
Relaxed.
Yeah.
And
Relaxed.
And Yang Huang is pushing very hard.
Very hard.
So I have a question I always want to ask you. So, you know, when you were a student, a graduate student at Tsinghua University, you start with very traditional topics of engineering mechanics.
Yes.
For example, the limit analysis, I remember it's for your master's thesis. Then you do fracture and then damage. And suddenly you change to biomechanics. Okay. So what are the special attractions of biomechanics that haunt you for so many years, and you are still doing this tirelessly? What the joy for doing biomechanics?
Yeah, that's a good question. As you know, when I worked in Germany, I also doing research on biomechanics and fracture mechanics with Professor Dietmar Gross. But at that time, I'm thinking about what I should do in the future. So that's the reason I moved to the Netherlands to work with Professor Eric van der Giesen, on nanocomposites. We studied on the nanomechanics. In 1999, I returned Tsinghua University as a third professor. And once I became a professor, I started to think that I should do something for the students. I should choose a good direction. The first direction I choose is soft materials. I studied the soft materials in 2000. So I got the national award the last year in the soft materials. We studied 20 years ago. As you know, 2006, you asked me to organize the Institute of Biomechanics. So if you don't ask me to do that, I think I don't move so fast to biomechanics. I think the first person who guided me in biomechanics is you, Professor Yang. And yeah.
So I know very little about biomechanics. I think Su Linzhang knew a lot about biomechanics. So, and I think today you choose the title of your presentation is the dynamics of a collective cells. So why you choose cells instead of organs, muscles, nerves, and so on?
Yeah, in fact, when Professor Zhejiang and Jimin Xia invited me give this talk, I gave them two topics. One is the soft tissues, and the topic which I got the national award last year. I studied the surface wrinkling, deformation of soft tissues by using continuum mechanics method. And the second topic I gave is the cells. And Zhigang and Jimmy told me that you can choose one, or you can give the abstract, and they can consider them. And finally, I looked through the past topics given by Professor Botoldi, Professor Alan Koo, and some others. I think they have talked too much on soft materials, soft tissues. So I think I should talk something different. That's why I chose the topic of cells. That's the reason.
Okay. And when did Zhigang approach you to persuade you to give a lecture, to give a webinar in this extreme mechanics letter webinar?
I think about
Jimmy
1 month.
contacted you.
1 month ago.
Yeah, Jimmy invited you.
Oh,
Yeah,
Jimmy. Okay.
Jimmy and Zhigang. Yeah. Yeah, they invited me. So I think
So
I should.
the face of Teng Li.
Of
Now
invitation.
you become a superstar in the wind chat, right?
Yeah.
So
Decision we made. Oh, excuse me. Decision we made. Invitation speaker. We make decision as a group. EML webinars, EML editors. We have periodically. We have emails around. People suggest names. Xi-Qiao, you have many friends.
Yeah, and in this talk, I mentioned that I used the entropy of Professor Constantino Salles, a very famous physical scientist in Brazil. And I think he will come to join this talk in a moment.
Oh, great. Okay.
He has been nominated. He has been nominated by several persons as a Nobel laureate. And I used the Salles entropy theory to explain the dynamics of collector cells. And
What's the name of this person?
Salles, T-S-A-L-L-I-S. But I think that he may be a little minutes later. Because
yeah, it's at the end of the lecture. That's entropy with a small case Q, right? Yeah.
Yeah. Yes, there's small Q. Q goes in.
Oh, wait. Some business item. I need to make sure you know how to use chat. There's a chat box.
I have never used chat before.
Okay. I know. You're a smart guy. You need to learn. At the bottom, there is a box called chat. Click it open.
So Zhigang said, do you see this message? Do you
Okay.
see this chat? Is
Yeah,
that one?
You reply, type in something. Type. Yeah. Sulin, you can ask Xi-Qiao some question.
I just promote Tin to the panelist. So this is a great library unit. I still remember that in old days, Xi-Qiao is our eldest graduate student and then postdoc in the lab, and Tin, Ten, and I were a graduate student there, play with them, you know.
So let me introduce Yuhang. Yuhang and I do not belong to Tsinghua University, right?
Okay.
we just went to better universities.
Okay.
You find very famous.
Yeah.
The University for the West Bridge.
Yuhang, can you please introduce yourself?
Yeah, Yuhang. I'm currently a colleague of Tin's. I'm in Georgia Tech in mechanical engineering and chemical and biomolecular engineering. Professor Feng and Yang, we met before. Professor Feng, we met several times in conference and also Tsinghua University. And but Professor Yang, we just met in distance in conference and places.
So
I'm more familiar with.
Before. Yeah.
Yeah, wait, let me ask you, let me do a little bit interview of you. Are you still Chinese Congress member now? Still?
No. We have actually two congresses. One is called Congress. Okay. I was a member of their standing committee say 3 years ago for 5 years. And now there's another one called Chinese People Political Consultative Committee Conference.
Oh, yeah.
Okay.
Okay. Yeah.
So I'm a member of that. Yeah. So actually today I was still in the meeting, but not for the evening. Okay.
Okay. Thank you.
We are currently discussing the 14th 5 years plan.
Yeah.
In this meeting. Yeah.
Thank you for doing this for EML webinar. I know how busy you are, and some years ago, I heard a terrific talk you give when you were a director or president or the Chinese. What is that organization? Chinese National Science Foundation. Do you call yourself a director or president? What do you guys call yourself?
President.
President.
Okay.
You are president of Zhejiang University. Your student, and also my student, Feng, insist Zhejiang University is the third best university in China.
Okay.
The third or second?
You know there's a joke for the university in China.
Yeah.
That means there are two universities. They all claim they are number one.
Yeah.
And there is no university claim they are number two. And many universities claim they are number three. Okay.
I see.
Do you know Feng will get married?
He is already married. We have a group. He sent even photos. We even got to know
the wedding will be the day after tomorrow.
No, they already had something yesterday. Yeah, there is a Qixi.
That's for the registration.
Oh, registration. All right.
Okay. And the photo.
Photos. Yeah, we already saw photos. And a little.
The day after tomorrow is the ceremony. I have to fly to Hangzhou to be the witness of their wedding.
Yeah. Unbelievable, beautiful people. Beautiful people.
Wei, and how are you?
Hey, Jimmy.
Hi, Jimmy. Yeah,
Good to
nice
see
to
you.
see you. Yeah.
Yeah,
Yeah.
Xi-Qiao, good to see
Yeah,
you.
yeah, thank you for your invitation.
Oh, thank you for doing this. This is wonderful. Yeah.
So many Tsinghua people. Unbelievable. Unbelievable.
Xi-Qiao.
Yeah,
How are
yeah,
you?
Ting Zhu, Su Lin, and we are very good friends for more than 20 years.
Oh yeah, I still remember we all work in that Xinjui's lab, right? 20
Yeah.
years ago.
So Ting, you are now in Georgia Tech.
Right. Yeah. Hi, Wei. Good evening.
So how is the situation there?
Our schools began 1 week ago, so we had some cluster spread over the weekend.
So the graduate students come back to the lab?
Right. We are
Okay.
like teaching in a hybrid mode. Some
Okay.
people teach online, some people teach in person, and some people combine the two teaching modes.
Okay, good.
So even the undergraduate students are coming back to campus.
Right. Most of them, yeah, came back. That's what I said. Then there was some cluster spread over the weekend. So our school took pretty aggressive testing measures. So they try to suppress those spreads.
Are there limits of number of students in the classroom? Beyond
Yes.
a certain number, you cannot have the in-person class.
Right. We reduced the capacity to 1/4 of the regular capacity in order to keep the social distancing.
Uh-huh.
Okay.
So
So
this,
between students, there should be at least 1 or 2 spacings in the seats, right? Yeah.
Right.
Yeah.
Yeah, we keep 6 feet social distance.
What about at Tsinghua or Zhejiang University? Are the classes normal now, resumed?
I think last semester we are all running classes in online, and so the graduate student of Zhejiang University get back in April, and so we are now many graduate students. We have group meetings every week. Which is a hybrid on the online and the offline and so on. So, and I think Tsinghua University, because there has been a rebound of the situation of in Beijing, so the undergraduate student didn't return to the campus for the previous semester, but they are coming to the campus now.
Yeah, probably we're young. I just noticed that today, and the number have reduced to 0 today.
Yeah.
It's clear.
Okay, and Xi-Qiao, I want to have a last question of my interview. Okay. And the question is, you have found an institute of biomechanics, and is that difficult to gathering something like 10 faculties in the institute, which is relatively new, and so how can you do that? Yeah.
Yes, in the past 10 years, we have recruited about 10 faculty members mainly from US. And because I think biomechanics in America is very excellent. It's much better than that in China. So we have many very young, very excellent young Chinese scholars in US, and I think it is a good chance for us to develop biomechanics in China. And now we have 3 presences with the Qingqian, including Bo Li, Li Bo, Shao Yue, and Zhao Zilong, Liu Chunjing, they are all Qingqian. So it's a very good chance to develop biomechanics in China, I think.
So Xi-Qiao, I have a question.
Zhao Zilong is the same name as the young general in the Three Kingdoms. Okay.
Yes, yes, well, completely the same. Today I will talk a little of his work.
Can I have a question to Xi-Qiao?
Yeah, sure.
Xi-Qiao, you know that in US, there is a big community, mechanobiology. How well have you connected to this community?
Yes, we have many friends there. For example, Ying Xiao, Wang Yingxiao, Fu Jianping, and so on. We are very good friends.
Great.
And several days ago, I discussed with Professor Wei Yang, I think. My point of view is that to do biomechanics, we must have very good foundations of mathematics, mechanics, and physics. If we don't have good foundations of mechanics, we cannot do good work in biomechanics. In China, some scholars doing research mainly on mechanobiology, they said the focus is on biology. I think it's good, but not enough, because if we do focus is on biology, what is all advantages over those in the school of biology science? I think our focus should be on the mechanics, on the modeling, theoretical analysis, and the analysis of mechanisms. That's all advantages over them.
Yeah, great.
Can I ask you to share your screen?
Yeah, okay, sure.
Yeah. So wait at 10 o'clock sharp, and you'll be in charge. You can formally introduce Xi-Qiao to the audience at 10 o'clock.
Okay.
So Xi-Qiao, maybe I can ask a short question here.
Yeah.
What is the main funding source of biomechanics in China? It's probably all related to Wei as well.
So I can partially answer this question. We are now making a big proposal for biomechanics, a block project right now. And so hopefully we can receive the funding and gather something about 50 to 100 people working on that. Yeah.
Well, that's great. That's great. The part of the reason I'm asking is I think that's I'm not really in the biomechanics research community in US, but that's my understanding is actually quite some challenge for biomechanics people in US to get the sustainable funding to really push that promising
The funding for biomechanics in China is in the division of medical science.
Medical science.
Yeah.
And recently in the NSFC have created a new department, 交叉学部.
Oh, I see. Okay.
Yeah, they can also support largely on the biomechanics, I think, in the future.
Okay, I think now is 10 o'clock p.m. in Beijing and a.m. In Boston. And hello, everyone. My name is Wei Yang. I'm currently a professor at Zhejiang University. And here I have the privilege to introduce Professor Xi-Qiao Feng. He is from the Institute of Biomechanics and Medical Engineering. That institute was founded by Professor Feng and his colleagues, and also in the Department of Engineering Mechanics. which was my previous department of Tsinghua University. Professor Feng is has working. He is an undergraduate and a master student and a PhD student. Received the 3 degrees all from Tsinghua University of Department of Engineering Mechanics. Actually, his PhD thesis was awarded the National Excellent PhD thesis. And he has spent some time in Germany and in Netherlands and also in United States to visit, and in the Germany as a Homburg Fellow. And Professor Feng is previously working on solid mechanics, for example, on fracture and damage mechanics. He is currently one of the editor-in-chief of Engineering Fracture Mechanics. And, but I think since about 10 or 15 years ago, he started working extensively in biomechanics and created this institute. And his work on soft materials and morphological change of soft materials has won him a National Natural Science Award last year, in the year 2019. And today he will give a new presentation on the new subject called the dynamics of collective cell. Without further ado, I'll give Professor Feng to start the presentation. Okay.
Yeah, thank you, Professor Yang. And first, I would like to thank Professor Yang for his nice introduction and for his greatest help to me in the past 30 years, more than 30 years. When I was an undergraduate student, Professor Wei Yang teaching us, and later he helped me really a lot. And ladies and gentlemen, good evening and good morning. First, I would like to thank Professor Jimmy Xia and Professor Zhigang Suo for inviting me to give this EML webinar talk. Today, I would like to discuss some of our recent works on the dynamics of collective cells. Most of these works have been done together with my colleagues and students. I will list their names in the end of this talk. And this is the outline of my talk. Following a brief introduction, I will discuss a few issues related to the mechanics of cells, which I think are interesting. They include our experimental and theoretical results on the differentiation, division, oscillation, and migration of cells. I will emphasize the physical mechanisms and the modeling of these dynamic processes. I think as a mechanics persons, we are good in modeling. In the introduction, I will explain why our particular attention is given to the scale of multiple collective cells. And if we want to study the cell mechanics, we found that it's so challenging because it's very complicated. And for mechanics, we need to consider 3 aspects. The first is force. In the cells, the forces the cells are subjected to have a very wide diversity of origins, many of which are as yet unclear. These forces may arise either from the interaction of the cells and their environment, or from the active process of the cell components, for example, the cytoskeleton membrane. And many of these forces acting on the micro scales are not deterministic. but they change a lot, and they exhibit the stochastic features, and their values are often difficult to be measured in experiments. Second, the cells have very complex type-specific and hierarchical structures consisting of a number of components, including the membrane, the nucleus, the cytoskeleton, cytoplasmas. Some of them are solid, some of them are fluid, which have greatly different mechanical, physical, biological properties and functions. All these structures, components, and their shapes and mechanical properties change a lot, change with time, and they change with space. Thirdly, most of the psychology and pathological processes involve not only one single processes of cells, but combined with various dynamic processes of cells, including the cell deformation, motion, adhesion, spreading, polarization, oscillation, aggregation, transmotion, proliferation, and so on. So it's very challenging. In the past years, we have spent some experimental and theoretical efforts to do research in cells, including stem cells and cancer cells in embryonic and tumor development. And our studies involve the molecular scales, some molecular scales, single cell scales, multiple cell scales, and also tissue scales and the organs and systems. But today, why our focus on the scales of multi-scale cells? This is because most of our physiological and pathological processes in our bodies involves the dynamic behaviors of interacting multiple cells instead of a single cell. And the study of the collective cells can bridge the micromechanics of cells and the continuum mechanics of tissues and organs. And at this scale, at the scale of multi-cells, we can find many novel and rich phenomena and mechanisms at this scale, which are greatly different from that in the cells and that in the tissues. I give a first example. This is the cellular embryonic tail bending. This bending is spontaneously. It's driven by two types of cells. That is the asymmetrical contractibility of no-charge cells and the cell proliferations of epithelial cells. In these two movies, we can see the dynamic behaviors of cells collectively. The first one is the oscillations, second one is the migration of cells. Another example is the tumor development. For example, the invasion and metastasis of cancer cells. In these processes, a large number of the cell clusters are involved. This is not a single cells can do this. They need to pass through a largely different tissues, for example, blood vessels, and they reach the organs where they can found a new tumor. And this is a real photo we got in the hospitals of bone cancer. The bone has already disappeared and the cancer cells migrate into the surrounding tissues. We can find that. And the second photo is the cell migrations in the lab. And in this talk, we'll discuss several typical dynamic behaviors of collective cells. The first one is the cell division. The second one is the cell migrations. The third one is the cell oscillations. In the recent years, we have made experimental and theoretical studies of collective cells, and we developed some experimental and theoretical methods to do research in this field. With aim of the following: we want to understand the generation and the transfer of forces at different length scales, and we want to understand how the collective cells cooperate each other to achieve their functions, and how the mechanical, biological, and chemical mechanisms are coupled. And also, we want to get some universal principles in the cell dynamic behaviors. And finally, we want to understand their physiological and pathological significance. Now let's consider the first part. This is the forces in the stem cell colony. This is an important tissue that we want to know how the cells can work together to generate forces, and how can they guarantee their integrity and of colony without any other supporting. And this is some photos of the growing embryo. The growing embryo. This is F-actin. The second column is F-actin, and the third is the E-catechin. And we can find their structures from these imaging figures. Now, if we put a large number of stem cells together, we can get a stem cell colony. But the stem cells can adhere each other, but the adhesion forces are not enough, in fact, to support the integrity of the spherical colony. So there must be some other forces to support the integrity. And we want to understand how these stem cells generate forces by themselves. And we got this is a cross section of the colony. And this is the shape and the geometry of cell nucleus. We observe the shapes of the cell nucleus. We found that they have different shapes. In the inner layer, the cells are nearly spherical, but outer layer, they are oblate. These shapes means that they have different forces. They are in different states of stresses. So we want to know where the forces are from, because we don't act any forces on them. And we found that it have a three-dimensional supercellular atomyosin cortex. We call it 3D suck structure. And we can see this movie we made. And it have such a network structures. It is hollow in the inside. It's hollow in the inside. And this network was formed by the stem cells themselves. And they form such a subcellular cap by the actin filament and myosin. In the network, it have tensile stresses. These stresses compresses the inner layer, such that the inner layer are subject to triaxial compressions, but the outer layers are subject to biaxial tension. So these forces generated by the cells guarantees the integrity of the colony and the stemness of the cells. This means that the cells can have the ability to different into different types of cells. So this is a new structures. Okay, and we studied the structures of this 3D suck, this F-actin, this myosin, and other components in structures. They combined into such a network. The mechanical and chemical couplings mechanisms generate forces where the self-assemblies of these different macromolecules into such a work. They generate forces for the stiffness of the structures. And we measured the magnitude of the surface tensions in the structures and the structures' mechanical properties and the stress regulations and different mechanical and biological functions in these things. And for more details, we can find them in this paper. Okay, the conclusion side, the collective cells can work together to generate forces at a larger scale than the cell itself. Now let's move to the second part, the force generation based cell division rules. And we must note that for cells, the force dictates geometry. This is important for us because the following reason. Okay. And in 1994, a German scientist, Hartwig, he proposed this very famous rule for the cell division. It's called long X laws of cell division. It states that the division plan of cells is perpendicular to the longest axis. The shape, that means that this is the shape center, and this is the long axis. So the cells will divide along the yellow lines, and this is true. But in many cases, these classical rules cannot explain the results in the experiments. The reason that these classical cell division rules is purely geometric. They have not considered any forces, any energy, and other mechanisms. But cells always love mechanics. They use mechanics to do things. This is our point of view. So let's consider cells in a confined space. These are cells. We consider different mechanisms involved in the processes. They include the cell substrate adhesion, the cell spreading, the dynamic cytoskeleton, geometric confinement, and with the elastic and the viscoelastic of cell membranes and cytoskeletons, and the motion of the nucleus. Okay. And I don't give the equations, but I give the results. This is a spreading of the cells, for example, these cytoskeletons, and we calculate the forces of the things. We can find that for such a cell, it has 3 extreme values, the red curve. But the maximal stresses, tensile stresses, is along these directions. This means that the nucleus is subjected to tension in these directions, the maximal stress. So we predict that the cells will divide along this line. And this is the force-based cell divisions we derived. And in fact, it is completely identical to the maximal tensile stresses criteria in the classical mechanics of materials for glasses, for concrete. This is called the first criteria of strength. And by using these rules, this rules is modified at this form. We see that the cell division plan is perpendicular to the maximal principal stress in this nucleus. It can be found that these rules can explain all of these results. Okay, this is the first step. Now let's into the second step. This new rule can explain the cell divisions in different conditions. For example, the cell division in these directions or in these directions. And in fact, a cell divisions in real tissues is very complicated, including the following 7 stages. 7 stages. this is cells, it's rounding, and so and we have many complicated processes. And this method can simulate the whole processes of cell division. And in these things, we should consider the mechanical, chemical, and biological factors, because all of these factors influences the forces, the magnitude and the directions of forces. And these forces acted in the cytoskeleton, cell nucleus, and finally dictate the division plan of the cells. So this is our basic protocols of our analysis. By this method, we can analyze the cell divisions in such a collective cell systems. For example, this is examples. The cells can rounding and rotation and finally divide along a certain directions. You can find the details in these very recent papers. And finally, I just give another interesting experiment. We consider the heating effect on cell division. And we use the cells, we give the cells a heat shock of 84 degrees for 10 minutes. And then they are incubated at the 37 degrees for long times, maybe 2 days, 3 days, or even longer. And it notes that some persons like the sauna. The sauna, the air temperatures is normally in the range of 75 degrees or even 100 degrees or even higher. And the time is much longer than 10 minutes. Okay, let's show the movie. The truth in the movie, but in the movies we have 2 main cells. Let's show a local regencies. We have 2 cells here. We can find some very surprising things. The cells, each cells was divided into not 2 cells, but divided into 3 cells. This is very surprising. This we call multipolar division. This, in fact, is a mistake. Such mistakes in your body may cause diseases, for example, cancer. And I think I don't want to tell the detailed mechanisms and why such things happen, such mistakes happens in the cells that lead to the multipolar deficiencies. And in fact, in recent days, I have been thinking about how we can get a theoretical model to study the multipolar deficiencies. I think, and I guess, the instabilities of the structures, including the cytoskeleton and the nucleus, might be a possible explanation, but this is a guess, this is not a conclusion. Now let's move to the oscillation of the cells. And we can show that in this movie, we can find this is a tissue of embryo tissue. You can find that the tissues oscillate with a periodic of 20 or 30 seconds, something like this. And why such things happened? We want to know the reason. The detailed equations can be found in this PNAS papers and this another paper. We consider these processes. In this stage, the cells oscillations, and in this stage, this is in fact is active seeping stage. We can use the fractal mechanics methods to study these processes by using the stress intensity factors to simulate the active processes of shaping of a embryo. But I don't talk this in this talk. Let's talk the oscillation. The tissue have such a shape, it have a large number of cells, and when perturbations is added in this position, for example, and these cells will subject to stretch, and the stretch will activate the biological processes to generate the F-actin and myosin. They can subject the forces to contract the cells to avoid its further stretching. And so they can deform the cells. And there is a cell stretch and chemical feedback between the chemical and the mechanical factors. And so they have different results. And this is the equation of the model. Let's show the results. So when the systems have a perturbation in the systems, it has three possible results, consequence. The first is the chemical mechanical equilibrium. This means that after a certain time, the systems will become stable. It cannot oscillate. But in some such conditions, the systems will reach hopeful bifurcations, have an oscillation state. This is caused by the hopeful bifurcations due to the mechanical and the chemical feedback. And there is a third possible. That means that the systems cannot subject such a large perturbation and it will be collapsed. So we have the phase diagrams in the three states. Okay, by using these models, we can simulate the oscillations of the tissue, and this is the numerical simulation results of our model. It found that it's very close to the experimental observations, and we can analyze the speed, directions, and other factors of these things. Okay, this is a very interesting topic of the oscillations of things. In these problems, the oscillations was caused by the mechanical and chemical feedback, and it induces the hopeful bifurcations, and the force induced in the cells themselves plays a very significant role. They trigger the oscillations, and it serves as a gating for the oscillations. And this model can be extended to other cell systems. Okay, let's move to the migration of collective cells. And to do this work, we first need to consider the platform. And in this work, we first set up a platform to study such things. Here are some examples of the collective rotation of cells, migration of cells. And for example, the two cells can rotate together into a Taiji pattern or a yin-yang pattern. And for another type of cells, the two cells cannot into such a shape, but it can rotate in such a manner like a tango dancing. And this is a simulation of Borley. And now let's show what we can do by using our experimental platform. For example, in such systems, we can put two types of cells or three types of cells. And this is one type of cells, and this is another one. And then we observe, we measure the velocities, the stresses, the shapes of each cell, and we get the date of these systems. Okay, and this is the movie, and this is the interface evolutions of one cells, and we can recount the deformations and of all the cells. And this is another cells. And so we can calculate the velocities of each cells. This is the results of our experiments. We can calculate all of the data, including the stresses and the velocities, energies, and we can calculate them. And this is the boundary between the two tissues, which evolves with time. And this is the stresses. And in this region, maybe we can have a larger shear stresses or compressive stresses. And this is the trajectories of each cells. They move from here to here, and we can follow each cell. And so we can get much information from this test. Okay, I do talk details of that things. Let's move to the migration. And this is one of our experimental results. We can find that all these cells are moved together, and we can recount them. You can find that these cells are rotated together, and here we can find another vertex, and they move at different patterns depending on the cell types and time. And this is the calculation result from this movie. Okay, now let's make a simulation of these things. Make a theoretical model. As I mentioned a little earlier, if we have different types of cells, they have different results. Why? Because they have different social interactions. And in some cases, the cells can adhere each other with very strong adhesion force. And one cells want to follow another cells, and we call it local alignment, or we call it big-cycle interactions. Another thing that the cells don't want to get in touch with another cells. They want to escape from each other. So we call it context inhibition of local motion. That's the reason we have different results of these different types. And we can give the brief equations. And this is the control equations. This is the potential forces, and this is polarizations. And this is the polarizations of a cell. It depends on the interactions of two cells, depending on the two parameters mu alpha and mu c, with the two social interaction mechanisms. And we can calculate elastic energies and so on. Now let's give the results. So by using these things, we can simulate the photo we just given in the last slides. We can define linear numbers for such cellular systems. The cells can swirl together, just like we have observed in the experiments. And if we put a certain number of cells in a circular region in a confined space, we can find that they have different modes of migration. When the cell is small, for example, we have 50 cells, they can rotate together. Just like a solid plate, they can rotate together. The velocities is proportional to the radius. But in this case, with increase of the cell, there's 100 cells, 200 cells, 500 cells. The outer layer, the cells cannot follow the inner layer because the inner layer have a small speed, but the outer layers must run very fast to follow them. And if they cannot follow them, they will lead to the formation of a vortex. Okay, these things just like the behaviors of a fluid, the fluidic mechanical behaviors. So we defined two parameters to describe such things. The first one is rotational order parameters. Second one is vortex numbers. We can find that they have two different stages. When the system is small, they rotate together. We call it the global rotation. But when the system is large, they have a local swirling pattern. Okay, by using this method, we can simulate any geometric shape. For example, this is in a channel, in a narrow channel, in wide channels, they have different results. I don't want to give the details of these results. And this method can also be used to simulate of cell deficiencies on the curved surfaces. This is just like the movie I shown a little earlier in the real embryo. And for example, we can simulate the dynamic behaviors of collected cells in such a large scales, which include several thousand or even more cells. We can calculate their dynamic behaviors by using this method. Now let's go to the statistics of cell migration. In this part, I would like to discuss how we should calculate the energy, the entropy, and the statistical laws of cell migration. We also want to get universal statistical laws for cell migrations because we don't have so much time and money to measure each type of cells because we have so many types of cells. They have different mechanical and properties. In addition, the dynamic behaviors of collected cells, depending on different conditions, they are much more complex. For example, we have very different types of cells. They have different mechanical properties and specific functions. The specific is the important feature of bio. Also, we can observe different dynamic phenomena in the experiments. For example, we can find different migration modes. In addition, the cells are located in different local environment. In the local environment, there are many factors, mechanical, physical, chemical factors, which all affect the cell adhesion, the ECM interactions. All of these things influencing the dynamic behaviors of collected cells. So we want to get the universal statistical law because there are so many factors. If we don't have such universal laws, we cannot make a prediction. So we want to establish universal statistical laws for collected cells. Now let's do this. First, let's do experiments to observe the large number of cells on a substrate. The substrate can have different stiffness. We measured different types of cells. For example, the MDCK cells. These are cells from a dog. This is human endothelial cells. This is the most myoblast cells. This is another embryo fibroblast cells. And we have any other types of cells. We want to get the universal statistical laws for all these cells and for different environmental factors. Okay, this is a two movies of two different types of the cells. This MDCK, this is HUVEC. You can find that the cells have different shapes and different properties, different behaviors. We want to get universal laws for them. We recount the positions and speed of each cells. And in this slide, we can have shown the moon directions of each cells. The scale bar is about 200 macrons. We can follow the paths and the trajectories of each cells. This is the cell nucleus trajectories. For example, these cells move from here to here and so on. Okay, let's make the statistical features of these things. And this is the mean speed of the cells, the average speed. And this is the variations, standard variations of the cells. They have nearly linear relationships. With time, the speed decreases along these lines. And the different colors means different times. These different times. Okay, but they follow this line. This is a jamming processes. Originally, they can move very fast, but finally they jam each other because of the loss of energy and speed. At different times, the starting the 3 hours, 6 hours, 9 hours, and 12 hours, we can find that they have different the probability density functions of the average speed. The average speed, this is our speed. And this is the velocity. We can find that with time increases, the speed decreases. But when they are scaled, they are normalized into a same curve. They are almost identical. This means that the scaled velocities is time invariant. Okay, so we can rule out the time mechanisms. For discrete systems, two conventional distributions are often used. The first one is Gauss distributions. They have such a form. Another one is called K-gamma distributions. It have such form. This K is the shape parameters, and this gamma is gamma function. And we want to know whether this classical functions works for all systems. Okay, this is the results. This is comparisons with these Gaussian distributions and the K-gamma functions. We can find that they cannot be used for all problems. This is the Gaussian distributions. They can only simulate this small range of velocity. And because in our test, they have a long tails, the Gauss distributions cannot simulate this curve. This means that this is not a Gauss distributions. And the K-gamma distributions is also widely used in the literature, but we found that in the K-gamma functions, it have such a stage, but in all systems, we don't get such a stage. So it is not a K-gamma function. So we must find another function to start these things. So it's to explain these results, we used a novel theory of entropy. It's called the Thales entropy. Professor Caetano Thales is a very famous Brazil physicist. I think he also listening this talk today. The Thales entropy is a very good generation of the classical Boltzmann-Gibbs entropy. It can be used to well describe the non-equilibrium systems with strong correlations among different microstates. Because in all systems, all the cells, they work together, they have a strong interaction and they have different social interactions. So it cannot be used, it cannot be described by the classical Boltzmann-Gibbs entropy, but maybe can be explained by the Salis entropy. The Salis entropy is defined in this form. And when in here we have a parameter Q, the Q is called the entropy index. When Q is approaching to 1, it can be reduced to the classical Boltzmann-Gibbs entropy in this form. So it is a generalization of the classical BG entropy. And if we use such a theory of Salis entropy, we can predict that the cell systems will follow another functions we called Q Gaussian distributions. Let's see the results. And this is the Q Gaussian distributions predicted by the Salis entropy. It has such form. And Q is the entropy index. We need to delimitate it from the experiments. Okay, this is the results. This is the comparison of our experimental data, a large number of data. It can be well described by the Q Gaussian distributions and by the Salis theory. And this is the equation. And the Q in the DEX, the Q index is experimentally identified as about 1.22 or 1.25 in this range. It changed only a little. Okay, so we can find this is a good functions for us. And this universal statistical laws works also for different types of cells. This is one type of cells, another one. You can find that they can also work very well. And so this universal statistical laws works not only for one single cells. It's also insensitive to the substrate stiffness. We can change the stiffness of the substrate. The results is not interfered. So the results is quite good by using this new theory of entropy. Okay, this is the comparison. This is the Gaussian, the classical Max Boltzmann distributions by the BG entropy. This is a Gaussian, the Salis entropy. You can find that the Q Gaussian distributions is much better. It can be used for all types of the different cells. So from this result, we found that for the cells, for dynamic behavior of the cells, they obey neither the Gaussian distributions or the K-gam distributions, which are often used in the literature. But for first times, we found that these cells should follow the Q Gaussian distributions. And this universal results works well for different types of cells, for different times, for different the local environmental parameters. And also we made experiments to show that the Q Gaussian distribution in fact stems from the cell-cell interactions. We can change the cell-cell interactions by using the biological methods, by changing by the chemical methods. Okay, and thus the Salis entropy theories provide us very good tools for studying the dynamic behaviors of cell systems. which have a strong correlation each other. For the results, we can, for details, we can find in this very new paper published just 1 week ago. And now I think I might give the conclusion. And in this talk, I have shown some of all recent works on the theoretical and theoretical, experimental and theoretical efforts toward understanding the dynamic behaviors of collective cells, including the division, differentiation, oscillation, migration. We want to understand them from the viewpoint of forces and energies. We want to understand them by using the knowledge of mechanics and physics. But our work is still in the very beginning stage, and we need to establish three-dimensional multi-scale model that can integrate the key microscopic mechanisms to predict the macroscopic dynamic behaviors. And we have students, they are doing this work. And also, our work should be more cooperated with the clinical issues, especially with the diagnosis and therapy of some diseases, such as cancer. And I think this is my talk. We have some publications, and finally, I want to thank my co-workers and the students. I have cooperated with Professor Huajian Gao for more than 20 years. And they helped us a lot in the biological knowledge. And also I want to thank Professor David Weiss. We have an international cooperation project sponsored by the National Science Foundation of China. And much of this work have been done by Dr. Bo Li. He guided my students to this, and also many other my colleagues and the students. And finally, I want to thank the National Science Foundation of China, and they supported these works, these fundamental works. And that's all. Thank you very much for your attention. Thank you.
Thank you. Hey, Xi-Qiao, can we ask you to unshare your window, your screen?
Yeah, okay.
So thank you, Xi-Qiao. Through about 50 minutes, you gave a very vivid presentation on the dynamics of a collective cell. And you emphasize the importance of force, the role it plays in how to gather the cell together, to decide the line of division, and to drive the migration of cells, and also the oscillation of cells through Hope bifurcation. At the end of the talk, you also talked about the statistical entropy, the solid statistic, the Q Gaussian. It's a wonderful presentation. So also there's two dense. You have about 90 slides presented in 15 minutes. So I think the audience should have a lot of questions. So how we start? We start with Jimmy. Anybody?
I have a question I'd like to ask.
Yeah, anyone who has questions can raise their hands. Then you can you can just call that person to ask.
Okay. Okay.
I have a question.
For example, David Weiss is now has raised his hand.
Oh.
Nan Shu has raised her hand. Can you see them?
And you see the blue hands at top right left corner.
Upper left corner of the little panel.
Of each individual's panel.
Okay, okay. So the first one is from Guy Jenny.
Hi, thanks so much. Xi-Qiao, it's a lot of fun going through 90 slides, 90 great slides in 50 great minutes. Thank you so much for this. So I'd like to ask a question on something all the way back on the Hopf bifurcation that you identified. And we discussed before that this is, and just so everyone knows, this is a huge, long-standing mystery that Xi-Qiao's group has uncovered. People have been talking about this since the 1970s. And one of the features that people have been talking about since the 1970s is that this sort of purse string shrinking down, it's really important to, this oscillatory shrinking down is important to development. It goes away in adulthood. We have these same cells with these same components in adulthood that these organisms in development that you so elegantly modeled have. What changes? Why is it that my whole body is not pulsating right now in adulthood? Whereas when I was the size of that organism that was in development, I was pulsating. What changes?
Do you mean the oscillation changes?
Yeah, so we switched from having this Hopf bifurcation that your group identified, and that has been talked about in the literature for decades. We switched from that to being stable. I am no longer pulsating as you look at me, or maybe I am and I can't tell. But what changes? What aspect, according to your model, changes to stop the oscillation with age?
Oh, okay. First, in the model, we have to contact to account for the interaction of the cells. The cell interactions have different mechanisms. They have the junctions, they have some slidings and many other actions. They are specific to the types of cells. So we have our modeling should account these mechanisms by mathematics equations. I think that is the changes. For example, the alignment and the inhibitions of the cells, we should give the appropriate equations for these things. And thus we can explain the dynamic behaviors and the different conditions and for the different types of cells. They have different parameters. In that model you mentioned, we have only 2 parameters that changes with the type of cells. And I think that is the key. The cells must be polarities in the cell. I did another point. For single cells, they move on substrate. If we don't only consider one cells, they move on substrate. They don't move along a straight line. They don't move like a Brown motion. They should follow a specific pathway. This pathway has been caused by the Levy flattening for aging pattern. And I think we should formulate this into the mathematical framework to get the real patterns for the cell dynamic behaviors, I think.
Okay, thank you, Xi-Qiao. I also saw several raising hand. One is from David Witz.
Yeah.
David, can you?
Thank you, Xi-Qiao. That was beautiful as always. Thank you very much for the talk. I certainly have learned I will never go to a sauna again. I don't want to see this bad division of my cells. But I have a sort of a more generic question for you. One of the beautiful things that you do are take these statistical approaches and describe the behavior, the phenomenological behavior. But of course, the real interesting thing, I think, is to understand something about the more fundamental aspects, the more fundamental physics aspects. So let me ask you a question about that I'm curious about. This beautiful example of the Thales entropy that you show so nicely fits all these data. Can you give me some physical intuition about what the underlying physics or physical chemistry that's causing this? What is it telling me other than the fact that it has this beautiful description of the behavior? Is there something more deep that we can learn? You know, for Maxwell Boltzmann, I sort of understand something of the equilibrium behavior. What do I really learn in this case? Is there something that we can say right away, or is it something that we still need to study?
Yeah, Dave, this is a really very good questions I have been thinking for a long time. And the Thales entropies can explain the statistics of discrete systems with strong correlation. Each is in the cells. The cells are active. They work together, and they have strong correlations. This cannot be explained by the classical Boltzmann entropy. And so the Thales, Professor Thales added another term that's called Q. This is the entropy index. But this index is determined in our labs by fitting the experimental results. And because the correlations of the cells are quite complex, they have different mechanics. And how can we correlate the value of Q with other parameters, for example, mu alpha, mu and mu L? And I don't know we have not got the detailed equations between these parameters. As you know, Professor Thales sent us 2 emails about these issues. They predict that for these systems we are studying, they predicted the alpha should be 1.25. But we have not calculated.
Professor Thales is on the screen, so may I ask him to
he's here. He's here.
comment?
Yeah. Maybe I think Professor Thales can answer these questions to Professor Weiss.
Are you hearing me right now?
Yes,
good.
we can hear you.
Yes.
Loud and clear.
Okay. Then first of all, I would like to thank Professor Feng for a beautiful talk on this very complex subject. Also, I would like to mention that by email a couple of days ago, I got contact with Professor Feng and his collaborators, and I suggested that in those experiments they could measure the anomalous diffusion of those motions, because the exponent of the anomalous diffusion would have a very simple connection with Q, in particular Q 1.2, that they measured in the Q Gaussians. And I think Professor Feng and collaborators might do these experiments, and that would be absolutely wonderful. Now, in what concerns the pertinent question by Professor Weitz, David Weitz, well, there is a lot of things that can be said about that, but basically Professor Feng gave you the key answer. When the elements of the system are very strongly correlated collectively among them, which is the case of many living organisms or living systems like languages, economics, and geophysics, earthquakes, many of those, then when the correlations are very strong, this forces the system not to use the entire phase space as was thought by Boltzmann and Gibbs and the traditional statistical mechanics. But those very strong correlations restrict the traveling within the phase space in a small part of it. Because of that, if you use Boltzmann-Gibbs entropy, it is not extensive anymore as it is required by thermodynamics. But if you adapt the value of Q to that particular system, you recover the thermodynamical extensivity of the entropy that we expect. It's a little bit analogous to the composition of velocities by Galileo. It is additive, right? V1 plus V2. But we know that it violates the Lorentz invariance that is present in Einstein relativity. So when you take into account the Lorentz invariance, you lose the Galilean additivity of velocity because it becomes a little bit more complex formula, which is V1 plus V2 divided by 1 plus, etc. So you lose additivity, but you gain the Lorentz invariance. Analogously here, by going from Boltzmann entropy to Q entropy, you lose the additivity of the entropy, but you gain something much more important, which is thermodynamical extensivity. Well, maybe this helps in this discussion. Thank you very much.
Oh, thank you, Professor Salis, and thank you for your comments. So I can see there are several people raising their hands. Let's give the microphone to the lady, Nanshu Lu.
Thank you, Professor Yang. Hello, Professor Feng, Zhejiang, everyone. This is Nanshu from University of Texas at Austin. I thoroughly enjoyed Professor Feng's extremely rich overview for cell mechanics. So I have 3 questions because it's a really complicated system. So Professor Feng, you mentioned many possible forces and cues acting on cells, right? For example, cell substrate interaction, cell-cell interaction, and internal cytoskeleton forces, and then chemical cues, bio cues. So I'm wondering if you can give us some numerical scales of those different forces. I'm asking because to me, right, after listening to your overview, you talked about like cell colony, cell division, cell oscillation and migration, and a lot of dynamics also. So I think for those different phenomena, there may be a few dominating forces. Maybe not all of those forces matter for those individual phenomena. So for example, right, the cell colony, I guess the surface tension and cell-cell interaction dominate. And for the division, cell division, the geometry and the cytoskeleton forces dominate, right? So how do we like have a more holistic understanding of those different phenomena based on the competition of those forces or energies?
Okay, thank you, Nanshu. This is a very good question. And at your site, I have discussed several different problems of cell dynamics. In these different problems, we have different equations. We have different dominant forces. This is the key to determine which forces are important for them. And for example, for the first part of cell division, we must calculate exactly the forces acted on the cell nucleus, which comes from the cell skeleton, which contact with the boundary with substrate. It also has some viscoelasticity, and it changes, it evolves, it rotates. So we must calculate all of these forces to determine the exact values of the forces. For these problems, it looks like not very complex, but in fact, you must calculate all of those forces in those systems, including the cell surface tension. But for some problems, for the migrations, we in fact don't consider too many forces. We consider the fraction between the cell and the substrate. We consider the interaction between the two cells. They have two different, we call social interactions of cells. And another part is the polarizations of cells. They change their directions, follow a certain equation. So this is the dominant forces. So I think how to choose the dominant forces depends on the problems what we are studying. Okay.
Thank you.
Actually, sorry, Professor Yang. So that's actually my next question.
Please be short. Okay.
Okay. So for like young researchers just starting cell mechanics research, do you have any advice for them on like how to choose or focus on the dominating forces? Because you have a lot of intuitions already, but for new researchers in this field, maybe they don't. And where to gain this kind of intuition?
This is very difficult problem for me. Yeah, as you know, we studied the biomechanics about 15 years ago, along with Professor Huadian Gao. And when I met him 15 years ago, he take a very thick textbooks of biology to study the biological knowledge, to understand the details of the cells. And then we can know what's important in different functions and different processes, and we can get the really important forces we need to calculate. So we must get some knowledge of real biological systems. So after that, we can get some intuitions to determine the forces. And
so with a big book, you can decide which one you
yeah.
So for all the people to raise questions, please first state your name and the institute. So I can see there's a raised hand by Brian Cox. And Brian, can you state the question? And the next one is Xi-Qiao Han. Okay, so Brian first.
Thank you very much. Good morning. I'm Brian Cox. I'm just me, no institution, deinstitutionalized, lost in the world. That was absolutely a beautiful talk, really very inspiring. So thank you for that. I'm fascinated by your story of cell division. How much do you understand about the mechanics of the nucleus itself and exactly how some force or strain information that the nucleus receives is transferred into the decision by the nucleus to divide itself?
Yes, this is a good question. And in fact, in my slides, we have a photo of the inner structures of a cell nucleus. We can observe the structural evolutions of cell nucleus at the nanoscales. And we can observe them. But in all models, we just simplify the nucleus as elastic containing bodies. We don't consider details, and we just determines the stress distributions along different directions, and then we decide the plan of division. Yeah, we don't consider details in the model of the nucleus. Thank you, Professor Cox.
Okay, and Xi-Qiao Han.
Thank you, Professor Yang. Xi-Qiao, that was beautiful talk, nice picture in the series. I have two kind of related questions. One is, in the model, do you treat the shear force versus tension, compression different or same? Another question is, in the model, you model the group of cells, how they move, interact. I guess you didn't consider the flow. So what if the flow come in, because the cell responds to flow, to the shear. If that come in, how would that affect the result, or how would you expect it affect?
Yeah, Professor Han. And yes, we have not considered the shear forces due to the blood flow. And because we did the test in the lab, we don't have any fluent flow. And so in the models, we have not considered this term, but we can act it. We can act it by adding the shear force. Another issue, I think, should be taken into account. If we consider the shear force, we should use the three-dimensional model. In this case, we are still constructing the three-dimensional dynamic models of cells. And
yeah, it would be great if you can include the shear, because, for example, in the blood vessel, the endothelial cell, they're under all this flow shear. Yeah. So it would be lots of interest to know how the cells migrate under this shear flow.
Yes, it's important. We have not considered the shear, but we can consider it in the three-dimensional model, I think.
Okay, so next we move to Yuhang Hu. I saw the raised hand.
Yeah. Thank you, Professor Yang. Thank you, Xi-Qiao, for the wonderful talk. It's really impressive. So I just have one question, but it has two folds. So first, I found it's often very difficult to work with soft materials and biological materials in general to quantify the material parameters in the model. So the question is, for this single cell model and the collective of cell behaviors, you didn't have time to talk about the details, but can you talk more details about what are the material parameters and how do you characterize those parameters that the values make sense? So that is the specific question. The other is through all this experience that you have studied many different phenomena and cell systems, do you have any suggestions of what parameters in the mechanics language, like adhesion or I think the stiffness and things like that, people have already measured. Anything that you would suggest the people to who are interested in characterizing the materials to look into the property that you think are critical in understanding the mechanics, but have not been really studied and characterized into details. Yeah.
Yeah. The first is the values of those parameters, especially some detailed values of the parameters in our model. In fact, it is quite difficult to experimentally measure the exact values of many parameters. We find some of the parameters in the literatures from which have been measured by some biological scientists. And but in fact, in the field of cell mechanics, I think many mechanical parameters in the classical mechanics of materials cannot be used directly, but these are still used. For example, I give example. For example, the Young's modulus. As you know, the Young's modulus was defined for continuum mechanics, which have been uniform, homogenized as uniform solid materials. But for the cells, it have a surface membranes in the including the cytoplasm and other structures. It's highly heterogeneous. In fact, I think the Young modulus cannot be used for cell. But in the literatures, we don't have a good parameters to define the mechanical properties of cell. So all the processes only use the Young's modulus of a cell because the Young's modulus of these cells is so much okay, and then we can use it. So if I think those parameters can be improved by our mechanics researchers in the future. And for the second, is how to make the modeling and how to use the mechanical languages to do that. I think I'm always a solid mechanics researchers. I just mainly use the languages of solid mechanics to do these things. I don't have much knowledge of biology. Despite of that, I have read some books, but I cannot remember those detailed words of biology. So I just want to use our knowledge of solid mechanics, fluid mechanics, and the dynamics into the cell. And that is all advantages to do research, to do biomechanics. As I mentioned a little earlier with Professor Wei Yang and Professor Zhejiang, in some professors in biomechanics, they just use the biological knowledge to do research. I think that is not enough. We must have solid foundations of mathematics, mechanics, and physics. That is all advantages of them. Physics is the important thing to do research of that.
Okay, so like Sally's statistics. Okay, I can see my colleague of Zhejiang University, Professor Ji Baohua Ji is raising hand. So Baohua, please.
Hello, Professor Yang. Thanks. Hello, everyone. Many thanks for Xi-Qiao's wonderful talk. Very impressed. You talk a lot of experiments and theories. Very impressed. On your collective cell rotation in the experiments on circular patterns. On large pattern, you show there are many vortex. That means there are low collectivity of cell rotation, right? On the small patterns, there are only one vortex. That means large collectivity. So does your experiment suggest a length scale for this kind of cooperation among the cells? Or there is a cell theory in your model to suggest this kind of length scale for the cell cooperation and coordination among this among those cells?
Yes, the characteristic size of the vertex is important parameters for the systems. It depends on the interactions of the cells, and also it changes with time. And in the simulations and also in the experiments, we can determine them. The value of the critical sizes. And but how to get an equation for that, we have not done that. The size is important. The size is important. 35.
Okay. Thank you.
Thank you.
Okay. So I also see a raised hand by Yaling Liu, and can you ask the question?
Hi, I'm Yang Lu from Lehigh University. Very wonderful talk, Professor Feng. So I think at the end of your slide, you were talking about some potential application in diagnosis and therapy. So that's why I was wondering whether have you tried, let's say, you show some interaction between cell that is have some factor about cell adhesion, and there is quite a few FDA-approved drugs that, say, for example, the sodium ATPase inhibitor that will reduce cell adhesion, and also there is chemokine that attract migration of cancer from one place to another place that triggers the metalysis. I was wondering, have you actively manipulate the environment or play with around the cell adhesion or the direct migration and see what happens?
Pardon, do you mean the entropy of the system?
The cell adhesion, let's say if you use some drug to reduce the cell adhesion,
okay.
What happens to their response?
Okay. The vertex and swirling patterns depends strongly on the interaction of the cells. You can using some chemical methods to change the interaction strength, intensities of cell-cell interactions, and thus it can change the patterns of the cell migration. And if the cell adhesion is very strong, the systems cannot rotate. It will be jammed in a jamming state. But if you wake the adhesion intensities, it can rotate much easier. It can have a very rich phenomena. Yeah.
Thank you.
In the biological labs, we have many methods to change those things. It's very easy.
Yeah, they might have therapy indication, right? And if you find like it reduce the migration, I think that would be helpful for reducing metalysis of cancer.
Yes, and the entropy, the solid entropy, can be used to calculate these things. But when they are scaled, when they are normalized, they almost independent on the adhesion. They independent on the specificity of the cell types, on the interactions between cell and the substrate and other parameters is universal statistical laws. Okay, Professor.
Wait, there are several of us that unfortunately are serving as host or co-host.
Yeah, I understand that. Yeah, I'll call you. Okay.
No. Wait, we don't have an option to raise our hand.
Yeah, I understand.
Okay.
You can send me a message. Okay.
Okay.
So maybe you ask the question first, then Zhejiang. Okay.
Oh, okay. All right. Thank you very much. Wei. Thank you, Xi-Qiao, for a wonderful talk. I'm Jimmy Xia. I'm from the Nanyang Technological University in Singapore. I'm also one of the co-editors in chief of Extreme Mechanics Letters, EML. I have two questions. One is for Professor Salas. The other one is for Xi-Qiao. The question for Professor Salas is, this is a beautiful, beautiful formulation, this Salas entropy. However, if you go back to the classical Boltzmann-Gibbs formulation, there's a formal formulation that would allow you to predict many behavior. For example, Gibbs free energy. You have a formal thermodynamics theory behind it, and you can predict many phenomena based on that. So my question is, with the Salas entropy, is there a corresponding formal formulation? If there is, how does it relate to thermodynamics? My question for Xi-Qiao is on the cell division part. I understand that you use mechanics, especially stress, particularly maximum principal stress, to make or study the cell division process. But the cell division process certainly is much more complex than just the stress-driven. Stress may play a role, and there's this spindle formation and all these other biological processes. The question is, how does stress and these biological processes interact with each other? Do you have something corresponding, or a stress value corresponding to certain biological processes? Maybe I'll ask Professor Salas to answer my first question and Xi-Qiao the second one.
Yeah, Professor Salas.
Professor Salas, can
Yes.
you answer the
Yes.
question? Yeah.
Yes. Are you hearing me?
Yep.
Yeah.
Yes. Okay. Yes. Of course, it's a very important point that our colleague raised. Indeed, the exponential of Boltzmann becomes a Q exponential, which is a slightly different form. When you take in the Q exponential, you take Q equal 1, you get Boltzmann exponential. The inverse function of the exponential is the logarithm. The inverse function of the Q exponential is the Q logarithm. So answering your question, the free energy, the thermodynamical free energy in normal statistical mechanics is minus kT logarithm of the partition function. And the partition function is a sum of exponentials. In the generalized version, you have the free energy being minus kT Q logarithm, instead of logarithm, Q logarithm of the Q partition function. And the Q partition function is a sum of Q exponentials instead of being a sum of exponentials. So all the thermodynamical Legendre transformation is preserved. It is Q generalized, and this has been published in 1991 with a Brazilian colleague, Curado, and it has been published in a more extended version in 1998 in Physica A from Elsevier, having as co-authors Professor Plastino from Argentina and Professor Mendez from Brazil. So yes, indeed, the entire Legendre structure of thermodynamics is Q invariant. And you have all the formulas are Q generalized. Well, that's
wonderful,
my answer.
Wonderful. Thank you very much.
Thanks, Professor Alice. Thanks for the very excellent explanation. So maybe Xi-Qiao, you answer the second part.
Yes, as Jimmy said, cell division is really very complicated processes. And in the textbooks of biology, they use the Hartwig rules proposed in 1884, that's 140 years ago. They just use the geometry to predict the cell division plan. But we want to put these rules one step forward. So we just use the classical solid mechanics method to predict the division plan. And we found that it is fun to predict the cell divisions in most cases we have studied. Since this process is very complicated, it involves a large number of different bio pieces, the force will activate some pathways of biology, including the molecular, the cytoskeleton, the nuclear, some things. So it's very, very complicated. But we don't consider those details. We just use the stress and the division plan. We want to get the relations between the two things. If more details, we can consider in the future, but I think it cannot be easily used in the future.
Yeah, I think Jamie can further elaborate this model. Okay. So maybe next we ask Zhejiang to ask a question.
Thank you. Thank you. Fascinating talk. So I have many specific questions, but I'll ask a general question. Now, of course, mechanicians all love things moving, and you show so many things moving simultaneously. Very complicated, very complicated. Therefore, very fascinating. But so traditionally, mechanicians work on fundamental things because there are some important applications. For example, when we worry about a fracture, it's because the ships break. And when we worry about a buckling, because the airplane wing is doing something. So maybe this is a question to you and other people who have thought about cell mechanics, such as David Weiss, the same question. So what do you guys think as application when you work with such a complicated mechanics? What is a vision? That's the question.
Okay.
Yes.
Applications. Okay.
Yeah, applications.
Potential
So maybe application now, maybe not ready, or other groups are doing this, but there must be some kind of collective vision within your field. I'm not in that field. What's the general application you guys are thinking about?
Yes, and in fact, we have cooperated with some hospitals for many years. And we have developed some medicines against the cancer. And we have some studies on the diagnosis of cancer. I just give, and some of the techniques, some results of my colleagues in my institute have been included in the handbook of Chinese medicine for diagnosis of cancer have been included in the handbooks, some colleagues. I just give one example. And in one kind of cancers, they observe some dot on the surfaces, some dot, periodic dot along one line or something like that. They don't know the reason. They don't know whether the dot indicates diseases or cancer or some other diseases. In fact, it's caused by the buckling of the blood vessels, by the stress-related growth, just like the Feng theory. The buckling of the vessels into the helical shapes, and give some prices on the skin, and finally the skins have some red dot. And by those periodic of the dot, we can analyze the underlying symptoms. In fact, these things can be used in the hospitals. We have some collaborations with some companies and hospitals. But today we only give some results on the microscopic cells, and we don't tell the detail, the contained mechanics results. Yeah.
Hey, David, can I ask you the same question? I know you have been thinking about cell mechanics, doing things. You always do things, not only for fun but also for something
useful
applications. What do you think about this?
I mean, I think it's a good question, Zhejiang. And the thing I find beautiful about what Xi-Qiao is telling us is I agree completely with him, as being a physicist myself, that we have to understand the physics, and he's doing that. I personally find it difficult to connect this to what biologists think about, because they really think at the molecular scale. Biology, interestingly enough, biology used to be a more macroscopic science. It's with sequencing and molecular biology, it's become increasingly molecular in scale. And my own view is that as physicists, we have to apply the physics, our physics way of thinking about things. But as scientists, we have to then relate that to the rest of the community and to always seek where can we learn something that, as you say, would be relevant to things beyond just the beauty of the science that we're doing. And to me, I think it's one of the biggest issues that we have. I will say, again, this is a very personal perspective, that there is a new evolving field in biology called mechanobiology. I think it's driven by work of the sort that Xi-Qiao is talking about. It needs more efforts of the sort that Xi-Qiao is talking about. I'm just delighted with all his recent stuff that he told us about the Salis entropy. I'd love to understand that more. But I think that there's, if anything, a huge opportunity now to look for something where what we learn from the beauty of, personally as a physicist or a mechanician, what we learn about that and to try and apply it to really understanding something that would be relevant for health or technology or something. If I knew how to do it better, Zhejiang, I would be doing it now myself. And I look to you and Xi-Qiao and others to help lead us in that direction. So it's a great issue.
So
another aspect.
Xi-Qiao has some patents. So the patent can be transferred and to sell for the money. And I think David just mentioned the mechanobiology. So I understand Sulin has working in this area for many years. Sulin, do you have questions or comments?
Sure. Thank you, Professor Yang. It's nice to see you online. And very rare. And Xi-Qiao, wonderful talk. I do have specific questions, but to answer Zhigang's question, I think that a lot of people develop this so-called mechanological interventions for cancer therapy, etc. So there are ways that you can make it use of mechanics to for cancer treatment, for disease treatment, stuff like that. So now I do have some concrete questions following up David Weiss and Jimmy. We know that, and this question also goes to Professor Tallis. So we know that for living systems, they constantly undergo this thermodynamically irreversible process. So by irreversibility, it means that it destroys orders near equilibrium and create or restore orders at far from equilibrium conditions. So you say that this living system constantly produce entropy or exchange entropy from with these surroundings. So how this is connected to your entropy-based universal fitting of the motion of the cells. So that's something that I wonder. Yeah, that's the first question. The second question is that an open question in biology is nucleation and growth, right? So we even don't know when we have eggs first or the chicken first, right? So for your spherical shaped cells or cell colonies, you say that you have fibronectin generated at the surrounding, and that will generate tension in this whole colony. So what caused the generation, or what caused the nucleation of this fibronectin aggregation? And what caused the first cause this generation of the tension, right? So that's two questions that I have.
Oh, very interesting questions. Let Professor Salis answer the first part. Professor Salis?
Yes. Basically, if you have a system that evolves in a phase space, which might be very, very complicated and multidimensional, if you take initially the system in a little part of the phase space and you leave it to evolve, it then travels and occupies further and further regions of phase space. While doing this, the entropy, Boltzmann entropy, is increasing with time. So it starts more or less from zero. It increases linearly with time. And finally, it saturates to a value which is close to the logarithm of W, the size of the phase space. So for normal systems, as a function of time, the entropy that increases linearly with time is precisely Boltzmann-Gibbs entropy. When you have a living organism or a living system, it is not so. The Boltzmann-Gibbs entropy does not increase linearly with time. It increases in a not very interesting shape. But if you change the value of Q, there is a special value of Q for which it increases linearly with time. That determines the value of Q. And the relaxation of that entropy towards its own equilibrium, it's not through an exponential relaxation. It's through a Q exponential relaxation, which has the same shape as the log-log figures that Professor Feng showed to us today. So in log-log is something like this, with a power law here. This connects to the point that was raised by Professor Weiss, referring to mechanical effects. And let me tell you, if you take a piece of cement of a building or a column or something, if you stress it, if you put a microphone, you will hear tick, 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 which are microfractures that appear inside the piece of cement or some part of a bridge. You don't see them, but you can hear them. And when you release, it stops. When you stress again you hear tick, 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 tick through the microphone. Those microfractures inside go further on along the previous microfractures. They just increase the microfractures that were already inside. When you make the statistics of these tick, 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 if you measure them with a computer, you make the statistics of the histograms that appears, they are Q exponential again, and they have a value of Q. And if you continue doing this, at a certain moment, the microfractures become a macrofracture, and the whole building goes down, it falls down. So by observing the acoustic emission of those little things that are happening inside, you can follow and you can even predict a little bit whether the building is stable or whether the building is in a dangerous situation, like the bridge that fell down in Genova, in Italy, a couple of years ago. So go into the systems that Professor Feng and Professor Weitz are analyzing. If there is something there that produces a sequence of little noises, tick, 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 something like that, if there is a phenomenon in those biosystems that behaves like this, if you analyze the histogram of those ticks, you can again obtain a Q exponential and relate it to the Q entropy. Thank you.
Okay, thank you. Xi-Qiao, could you answer the second part of Louis' question?
What is the second part of the question?
Thank you, Sulin. As the chicken and egg relation. The
chicken or egg? Okay.
Egg. And I think this is a good question. A cell is a very smart system. It has many sensors in the cell, especially on the surfaces, on the cell membranes. The membranes is very complicated structures. They have many sensors and many air channels. The sensors can sense temperature, can sense the forces, can sense the stresses, can sense many other stimuli. And then they can activate some biological processes to inside, to pass away the signal inside and to activate some things. Therefore, the inner structures, including the cytoskeletons and some other molecules, will accumulate each other to work together to respond to the external stimuli and also to adapt to the variations of the environmental. So I think this is the chicken and the egg. They have sensors to determine what they should do to adapt to the environment. This is one thing. Another thing I think I want to emphasize that the cell is very complicated. We cannot predict its behaviors only from the knowledge of solid mechanics. In fact, if we predict some things from the mechanical of materials, you found that many things cannot be explained because you often neglect some important things which cannot be predicted things. And for example, I give you an example for the adhesion. We know that on the surfaces of the cell membranes, they have some integrin molecules. In most of the models, they think that the molecules spread and they can diffuse on the surface of the cell membranes, and then they calculate the adhesion forces. You mentioned adhesion, okay? But in fact, the number of those integrins are not constant. They change a lot, and the integrins can be activated or not. They can be introcyted into the cells. They can pass through the cell membranes, and thus it cannot be assumed to be a constant. That is the complexities of cells. So we must do many tests, many experiments to get the processes. We cannot only imagine the processes by our brain. Thank you.
Thank you, Xi-Qiao. I saw there's a message from Professor Tian Tang. It's from University of Alberta. So it's a nice lady. So Tian, could you ask the question?
Yes, thank you, Professor Yang, and thank you, Professor Feng. I had a bit of problem figuring out how to raise my hand. I raised my hand when I was not promoted, and I thought I was in the queue, and then I just learned from Nanshu how to raise the hand as a panelist. So thank you very much, Professor Yang and Professor Feng, for the extremely fascinating and rich talk. I learned a lot, and I think I would dig out some of the references that you were mentioning to learn more. I have many questions, but I want to ask one about the ECM stiffness, the effect of the stiffness of the substrate. A couple of years ago, we looked at how two bacteria colonies interact, and we found that there is a very significant effect from the stiffness of the substrate. And depending on the stiffness, the bacteria colonies may merge into a single one, or they could stay separated and with a clear interface between them. And what we argued was the rate of diffusion of those bacteria, which depends on the stiffness of the substrate. And in your presentation, you showed the velocity distribution, and you were able to nicely collapse all the curves into a single one. So I'm wondering if you could elaborate on how the ECM stiffness affect the distribution of the velocities and how you were able to collapse them into a single curve. Thank you.
Okay, Xi-Qiao.
The stiffness of substrate and have a big influences on the dynamic behaviors of cells on them. On one hand, we can change the adhesion interface it changes a lot. But if they are normalized by using the Salis therapy, it can be unified into universal laws. That's our results. Another point I want to emphasize that we use the Young's modulus of substrate. We often use some PDMS as a substrate, but in fact, the Young's modulus is not enough as to determine the results. I think Professor David Letts can give some answer because he have a very good work recently to say that the microstructures of substrate have a big influences on the results. Steve, can you explain that?
Can you elaborate on that?
Zongyuan, the Zongyuan's work with you.
You know, I'm afraid that you're leaving me a little blank, Xi-Qiao. I was trying to answer a phone at the same time. I apologize. So we do a lot of work on substrate structures, and so I'm not sure exactly which one you're meaning. Could you give me a little clue, since you're putting me on the spot? I apologize.
Okay, I guess I said that, and they compared, and Zongyuan and Jingxia, they correct the work.
Right. Okay.
And if the Young's modulus of the two substrates are same, but they have different porosity. For example, the first examples have large porosities, but small numbers of porosities. Another one have changes in the porosities, but the Young's modulus are same, but the results are greatly different. So if you want to do some tests on those substrates, they have a big effect. And I don't know what.
Yeah, no, Xi-Qiao, maybe I could just fill that in slightly, and that is you were talking about Jingxia's work, and he does really amazing work. A student from Tsinghua, right? So he must be great. But he's also now a student in my group who just graduated. Anybody looking for a great postdoc should talk to him. But he has very, very cleverly tried to distinguish, as a physicist or a mechanician, different aspects of physical structures. For example, if you think of a hydrogel, one normally thinks as the mesh size is completely correlated with the stiffness. But you might ask, well, if you could change the stiffness independently of the mesh size, would you change the way molecular interactions occur, because molecules can diffuse, nutrients can diffuse differently, but then have the stiffness controlled? And so he came up with a very clever way, again, following on Xi-Qiao's great contributions, to change the effective modulus by using layered structures. And that could allow him to distinguish pore size or mesh size and mechanical properties. And I think, to me, that's just one example of the type of thing that one has to do as physicists or as mechanicians, as mechanical engineers, to think, to address the kind of questions that you're really asking. That is, what are the key aspects? Let's not just think we have a system that's given to us, but let's try to control it in a way that we can understand and tease out the differences between the different contributions. I think this is what Xi-Qiao is quite correctly pointing out, that there's a tremendous opportunity for important studies to do that.
Okay, thank you, David. I thought Professor Teng Li has a question of University of Maryland.
Yeah, this is Teng Li from University of Maryland, and I'm also one of the associate editor of Mechanics Letters. Thanks, Xi-Qiao, for this wonderful and very rich talk, a lot of materials to digest. I have a general question, I guess not only for you, but also for our audience, whoever working on the cell mechanics. So in the biological system, for example, a human body, we do have some cells, they are in the two-dimensional nature, like the epithelial cells in our skin, the surface of the organ. But the majority part of the cells are actually in 3D nature. When you do the research in the lab, you start with a rather two-dimensional environment, right? You grow and cultivate cells in a petri dish, and you do the observation of the how cell divide, how they grow, how they invade, for example, the cancer cells into the healthy cells. But in reality, for example, a cancer cell start to divide and start to grow into a tumor. It's most likely in the three-dimensional nature. Now, I do see in your last slide that you are talking about the future plan. You are gonna look into the three-dimensional model of these cell collective behavior dynamics. So my question is, now, what will be the most important couple of mechanism or parameters that is not captured by the two-dimensional model or study, but could be important or even crucial to understand the actual three-dimensional behavior of the cells or collective cells? Thank you.
Okay, Tom, this is a very good question. First, we studied two-dimensional cell model layer because it's simple. It's very easy for us to make the test, the shape of the cells, the velocity, and the dynamic behaviors, and we can get the exact values of the cell, each cells. But in three-dimensional problems, it's much more complicated to make the experiments. And in the literature, some persons have measured the three-dimensional problems, including only 1 or 2 or 3 cells. This cannot be easily to do tests of so many cells. That is one reason. And in fact, we have one PhD student. He has established a well-done three-dimensional model, including a number of cells embedded or aligned on a substrate or embedded in an ECM to simulate the behaviors. It is a quite complicated numerical model, but we have not done tests in the result. So we have not present the three-dimensional results. That's the reason. Yeah, we can do that. Later, I can show you some slides after the talk. Okay.
Okay. So you have further results for the three-dimensional case.
Yeah. Yeah.
Okay. I also saw my colleague, Professor Zheng Jia of Zhejiang University to raise the hand. So Zheng, could you ask the question?
Yeah, thanks, Professor Yang, for the introduction. Professor Feng, thanks for the fantastic talk. This is Zheng Jia from Zhejiang University. So I would like to ask a question about the combination of cells and soft materials. So we know many soft materials can deform in response to external stimuli, so that we call them as smart soft materials. Also, we know cells are more than smart. So cells are intelligent because they can sense many different kinds of stimuli in the environment, and so they can self-proliferate and they can self-propagate. They can generate energy by themselves. Since you are also an expert on soft materials, so I would like to ask you a question about, so in the future, is that possible to combine cells and soft materials to make intelligent soft materials or intelligent soft robotics? That's my question. Thank you.
This is a really good question. In fact, in China, several related projects have been supported by the Department of Science and Technologies. In the Shenyang, there is an institute of CAS. They have been doing this. They combine the smart cells in soft materials and also some devices, and ask let the cells drive the motion of the device. This is a very frontier topic, I think. And if like I can show you some slides. They give us some slides because I was asked to be a committee members of those projects of CAS.
Thank you, Professor Feng. Yeah, thank you.
Recently there is a very good papers in PNAS on this topic, you can find it in 2019. Okay.
Yeah, thank you.
Okay. Is there a question from Changqing Sun? Hello. If not, I may ask a question. Okay. So at the very beginning of your presentation, you gave a formula, the second law of Newton. That is force equal to mass times acceleration. And you claim what can force represent, what mass represent, what acceleration represents. But in your dynamics, the collective cell dynamics, do you really consider the inertia of mass? That's my question.
Yeah, Professor Yang. In the simulations, in fact, we don't consider that part of energy because the initial is very, the speed, in fact, is very slow, and that part is not important, I think. And in the first page of my slide, I use M to stand for the constitutive behaviors of the structure of the material itself. And the structure is mechanical behaviors and the other the structure evolutions and those things. That I mean is that the properties of the cell itself. So we don't consider the initial energy.
And also in the presentation on the migration of collective cells, what is the driving force for the migration?
The driving force, if you put a cell on a table, the cell is the living. It always wants to find some food.
Okay.
Also, it's always walk around. So each cell want to walk around on the table, and finally they want to save energy, and finally they form some patterns. So they have they use
in the form of energetic force.
Yeah, they have they are living. They are like presences. We don't want to stand here. We want to look around to find something interesting.
Okay.
And somebody thinks that, and the migration of each cell should follow a specific pace. It's not random. It should follow the Levy flight for aging pace. Just like the path of bacteria, the path of a fly, a pattern of a fish, a pattern of a bird in the sky, then it's not random. Yeah, they have some similarities.
So that means the cancer and the food, if they combine together, mix together, has better energy than if they are separate.
Yes. Yes. Yeah. In fact, if you support, you provide sufficient nutrient to the tumors, and if it don't occur at a critical position in your body, it's safe. Why not?
So just want to make a comment here for Professor Yang's question. And there indeed, I heard there indeed some therapy, cancer therapy called the starvation. Don't provide any food to cancer cells, so that so don't divide. They don't have energy to divide, etc.
Yeah.
Yeah.
Sure.
So following that question, so I found a difficulty in developing models from mechanics point of view is that So we know that living matters or living materials live through constant flow of materials and energy. So our mechanicians, most of the time, only consider one kind of energy, that's mechanical energy. So we have difficulties to take into account or incorporate other energy forms into our formula. I wonder if Xi-Qiao have felt the same kind of difficulties, and how would we deal with that? And I think this is a discussion question, and I would like to have some comments from Professor David Weiss as well.
Before you answer that question, may I suggest that we shorten today's question and answer session slightly, because it's 12:15, 12 meaning midnight in Beijing. But you know, Wei Yang and Xi-Qiao, they are in the wee hours, including myself. So maybe because of that, let's try to shorten it.
Okay. So okay. we are stopped at 12:30, before
Okay.
12:30. Okay. So Xi-Qiao, could you comment on that?
Yes, I think students have raised a critical question. In fact, in the field of biomechanics, many persons want to account for other energies, including the biological related energy, the nutrient, the transport of nutrient, water, and other the biological related energy, including the chemical energy in the part, and including some different phases of the materials, the solid materials, liquid materials, and so on. We also published a few papers on the modelings, including the cell and including the biomechanical and chemical energies in the systems. And if we want to stimulate from the viewpoint of continuum mechanics, we must consider the nutrient transport, the water transport, and the waste transport, and so on, which we should consider the thermodynamic equilibrium for the things. In fact, we have some publications I can show you. We have 2 or 3 papers in JMS in the past 2 years on the chemical part of energy.
So I have
So can I just make one comment about that? Sorry, I'll be very brief. I would maybe look at it rather than the glass half empty, the glass half full. And my view of what Xi-Qiao has done is he's shown how you can take so many things and put them all together, understand something about the mechanics. And the beauty of it, in my view, is that he doesn't get lost in the details of all the other things. And the counterpoint would be that, you know, if you start looking at all the sequence events and all these things, there's enormous amount of detail, but you lose the really elegant generality and simplicity that Xi-Qiao shows. And to me, that adds first, some elegance, and second, it gives me ways of thinking about it, even just thinking about the mechanics and sort of taking all the other things and putting them together, but still understanding something by simplifying it to something very visible, something measurable, something tangible. So I would, while I agree completely with what you say, I would sort of look at it as tremendous progress and a tremendous advantage. And again, this is exactly the sort of thing that somebody like a mechanician like Xi-Qiao can do that a biologist who takes a different approach, also very valid, will not do. So I think it's tremendous, the sort of thing that we heard today.
Thank you, David. You gave me a lot of confidence. Thanks, Xi-Qiao, for your answer.
So Xi-Qiao, I have another question.
Yeah.
You tried different stimulus for the cell. You tried the thermal, and you tried the chemical. Have you tried think about the optical, the light, the laser beam, and so on, to stimulate the?
No, we have not considered optical stimulus. In the test, in one test, we used the laser to do the test, but we didn't to measure the detailed response to optical stimuli. Not yet.
Okay.
Not yet.
So, any more questions and comments?
I think Teng has a question.
Teng?
Ting Zhu.
No, Ting.
Yes. Yeah. Hi, this is Ting from Georgia Tech. Yeah, Xi-Qiao is really a wonderful talk. But I have some very fundamental questions. In your talk, I noticed that most of the time you use the term of force rather than the stress. Also, you seldom talk about the stress-strain relation or the constitutive model you use. So let's say when we think about the stress, I think we probably have different meanings when we take different representative volume elements. It's at a multicellular level or at a subcellular level. So let's say if we look at the stress at the subcellular level, we must recognize the highly heterogeneous microstructure, micro nanofibers inside the cells. So I wonder, and how did you account for those effects, and what kind of stress-strain relations did you use? I can draw an analogy to hard materials like polycrystalline metals and alloys, because I've been working on that for many years and very familiar with that. So we know under many, and also there are similar heterogeneous cell structures within grains. But those cells are the dislocation cells. They are also heterogeneous structure. They organize in a highly heterogeneous fashion in space. So as a result, they have a dramatic effect on the stress-strain behavior. For example, they generate the internal stress, which is very important. Nowadays, people can use X-ray synchrotron to measure those internal stresses. They can be they also govern this like kinematic hardening, asymmetry of stress-strain behavior. I would imagine there are also similar effects for this subcellular stress-strain relation, if you really consider the impact of this heterogeneous microstructures. Certainly cells are active, they have other roles, and I mean, they are more complex. So can you comment on your understanding of a stress at the subcellular level, and what kind of a stress-strain relation would you use, and how would you develop more physically based models and that really are measurable and can be used to really make more predictive simulations rather than just fitting the experiments? Yeah, that's my question.
Yes, Tim. Indeed, my point of view is that at the scale of subcellular, if we want to calculate the structural evolutions, I prefer to use the concept of force rather than the stresses, because in each problems, the key difficulties is to determine what forces are the real reasons for the response. It's not the stresses. Sometimes I use the stresses for example, in the cell deficiencies. In fact, it is not a real stress. It is still a force acted on the cell nucleus. In that models, we have averages the forces along the angular direction, So I prefer to use the concept of the force. In these studies, we want to make some simple models to understand the physics of this phenomena, rather than the real quantifications. We don't want to say that this value is 1.2, that value is 2.3. I think that is not important for us to understand. We want to know the physics, basic physics, the fundamental knowledges of these things. I think this is the first step for us to do research.
Okay.
Can I add something to that? Wait.
Very brief. Okay.
Very brief, right. So in the cellular mechanics, especially intracellular phenomena, all these things are non-local.
Yeah.
Many of these things are non-local. So
Okay.
the concept of stress does not always apply.
Okay. Yeah. So you mean force is more important?
Absolutely.
Okay. So I saw a raised hand by Changyong Cao. Changyong, could you ask the question very briefly, because we are close to 12:30.
Hello. Can you hear me? Thank you, Professor Xiqiao. Thank you, Professor Feng, for the wonderful talk. Professor, I'm Changyong Cao from Michigan State University. Actually, I want to ask a specific question because before, when I worked with Xuanhe, we did some work on the stem cell substrate interactions. We collaborated with Professor Cam Liang from Columbia. So we at that moment we know, it's really the stem cells is really smart, and we use the dynamic substrate topologies, and it's when we have the patterned surface, it will alignment well, and if we get it the flattening again, they spread randomly distributed. So and after if we change the pattern, it will form alignment again. So it's really how strong the influence for the cultural environment. So Professor Feng, you show a very beautiful, elegant, the statistical law. So I'm wondering if place the cells 3T3 or MDCK, these kind of cells, it will be also have effect, or is the how it affect your the statistical law.
Have you got the question?
Yeah, I somewhat I got the question. Is the culture environment has influences on the statistics or not? In fact, we have cultured the cell, the different types of cells on different substrate and different conditions. The speed, the absolute values of the speed changes a lot, but static universal laws can be scaled into an identical curve. It's no problem.
Okay.
Okay, I think now time is 12:30. There's still some raised hand, but I think it's already here. It's half hour past midnight, and thank you very much for the stimulate discussions, and we'll also thank Professor Feng for his very interesting talks on the collective cell dynamics. So back to Zhejiang. Okay.
Oh, I have nothing to. Thank you very much, Wei, for spending time with us. I know how busy you are. Thank you. Really appreciate it. All right.
Like that. Okay. Round of applause.
Thank you,
Thank you,
Yang, Jimmy, Dave. Thank you very much. Thank you all. Yeah, thank you all. Yeah.
Thank you, Xi-Qiao.
Yeah, Dave. I'm looking to see you again, Dave.
Yeah, me too. I hope so soon.
Thank you so much.
Bye.
Bye.
Yeah.
Bye.
Bye.
Yeah, Zhigang, thank you.
Thank you. Thank you. You must be tired.
Okay.
Typical. Typical. All right. So do send us suggestions of future speakers.
Yeah.
Yeah. So we'll put on our list. We have a long list, but every suggestion is on the list. So, and then editors will discuss. We'll try to maintain diversity and try to stabilize the platform, all these random considerations. At the end, we need to make decision. There's a speaker of the week.
So Zhigang,
that's random.
so Zhigang, Xi-Qiao will send us a write-up for 3 pages for this webinar, right?
Yeah. He promised to do that.
Yeah. I'll send you within 1 week.
Yeah,
Okay.
within 1 week. You can actually send to Sulin. You don't need to send Sulin, as long as he follows the format. And if you have time, take a look. There's no need for external review, right? For this kind of thing.
Yeah.
It's you're already invited.
Yeah.
And I submit it in with the platform of the journal. Yes.
Yeah. Yeah.
Okay.
Yeah. As you submit, just take a look at the format. I send the email to you. Very simple.
Okay. Okay. No problem.
So I guess Xi-Qiao, when you submit, you can choose specific editors. So if you choose me, I'll take a quick look, and then I'll accept them in no time. You know.
Yeah.
Okay.
Yeah.
Okay. Thank you. Thank you.
Thank you.
Yeah. Have great sleep.
Yeah.
Bye bye.
Bye. Bye. Bye.